All right, good afternoon, everyone. Um, so my talk would be a little bit different than what you've been hearing all day. It's basically looking at the wonderful work that started with Dr. Kang's lab and all the wonderful data you're seeing with Dr. Greenspan. The question comes in, what's next? How do we take this data and make it into a therapy that patients can access? So we're a group of what I call drug hunters, drug discovery people. I'm from the pharma industry. We're a group that got together and say, how can we get into an area where we can make therapeutics affordable and accessible to patients? So where we got involved is the idea to say, how do we make drugs accessible and affordable? That's the main goal for IM Therapeutics. Briefly, SLC6A1 and STXBP1 surely are the two diseases we're talking together, but there are over 9,000 identified rare diseases now, and it's counting. And if you imagine the time and the money it would take to find drugs for each one of those, I think we would be waiting a few generations and perhaps having to come up with trillions of dollars to make it happen. And the sad reality is that, the availability of drugs, accessibility of drugs, even in the US, is very hard. I would even venture out to say that most of us are one serious disease away from bankruptcy. And so in that situation, I think repurposing could be a great idea, because these are drugs that have been in use in humans. They've established to be safe and tolerated. All we are doing is finding new indications. The end of the story, though, is we have to make a commercial case for this to happen, because somebody has to eventually pick up this drug, make it commercially available, and invest in this testing for this new indication. And that's where this whole repurposing process has stalled. Otherwise, it would be great, and it has served the pharmaceutical industry very well, because the NCEs continue to be the way to go, which costs huge amounts of money and take huge amounts of time and money to develop. So this is where it comes in, where the cost of therapeutics, based on exclusivity, to ensure profitability. At the end, pharma companies are public companies. They have fiduciary responsibility to their shareholders. And they have to generate the profits that their shareholders are looking for. And the model has been built on blockbuster drugs that don't sell more than $2 billion in third year of launch, often don't even get the approval internally in pharma industry to be developed. And orphan drug designation law was put in place so that pharma industry can focus on addressing diseases that don't have huge addressable markets, but are limited to small population. But that has become almost a source for high expensive drugs for the pharma industry, where the average cost per drug is about $200,000 per year. I mean, Ravicti clearly is an outlier, but the price still is pretty daunting. And of course, priority review vouchers is one thing that the pharma industry likes. They don't get it if you repurpose a drug. So the way you do it is you do an NCE for a orphan indication, and you are eligible for a priority review voucher. Well, of course, all that equates to what we are seeing with Ravicti at $740,000. So what we are doing is really trying to build a collaborative model for R&D where we can take and address these issues, such as create an NCE from a repurposed drug, still look at it from a process where an experienced team can take this forward. What we rely on are the beautiful scientific data that is being generated in academic labs, such as Dr. Kang's lab, working with clinicians such as Dr. Greenspan and Dr. Demarest and Dr. Goodspeed, to really understand the clinical population, understand the science, and do the translation so that the therapeutic that comes out has a safety profile that the FDA would accept. Often we focus so much on the pharmacology of a drug it's the safety profile of the drug that really carries the day with the FDA. And often 90% of the time we spend is worrying about the safety profile of the drug. Because the first thing the drug has to do is not harm the patient before it can help the patient. So what we're really creating is these multilateral partnerships, such as the foundations, advocacy groups, the clinicians, researchers, impact investors, et cetera, to really come up with an idea how can we advance these therapeutics. Because as you will appreciate, potency, efficacy is clearly one hallmark of a drug, but all these other properties that have to be addressed before a inhibitor or something that works in an animal model can work in a drug. And these are empirical issues. There's really no a priori assays that you can do. You simply have to go through the process, anticipate what the issues could be, and figure out how to address them. And it relates to drug distribution, metabolism, formulation, manufacturing, etc. And this is where we come together as a team and say, how can we take something like phenylbutyric acid and turn this into a drug? So I'm going to skip this for the time, but this really is a global exercise. So we have now assembled these entities all over the world, between CROs that do the toxicology, CROs that do manufacturing, CROs that can do the formulation for us, metabolite ID, so we'll go to Poland, depending on where this can be done cost effectively and well. Because the main idea here is that if you can reduce the burden on the cost development of the drug, there is a potential that you can sell it at a price where you can still make a business case for it. Now, pharma says it takes 13 years and over $2 billion to get an NCE through. If that is the marker, then of course affordability will always be a challenge, but I think we can change that. Now, in terms of the science, and I'll talk a little bit about this, but you've heard enough about Ravicti, you've heard enough about what it does, so I don't need to belabor the point. But one of the points that Dr. Greenspan raised up was, how does it work? It's a very good question, because if we understand how it works, we can actually perhaps come up with a drug that is better than phenylbutyrate, because it still requires 7 to 10 grams a day. That's a hefty dose burden on patients. And while we've looked at it for a year, we don't know what happens 5 years down the road. So we have to be prepared. And the more you reduce the dose burden on a patient, the side effect profile can be managed. Because when you give such a whopping dose, there is an issue that can play out down the road. We don't know. So in that context, first we came up with a drug called ATX-018. The only difference between Ravicti and ATX-018 is ATX-018, it sits on an erythritol backbone rather than a glycerol backbone. So it still sits on a very safe backbone like erythritol, as it releases phenylbutyric acid. But the main thing is you can give 25% less compared to Ravicti. Not a big difference, but moving in the right direction. At least we can go. And fortunately, we own the composition of matter patent. And if we build a business case that we can develop it for a lot less than what normal pharma developments take, we could probably price it better. In this slide, I give you a little bit about what is known about potential improvements on side effects that can happen. I know Ravicti is still very well tolerated, but glycerol still has dose-limiting toxicities that can be there, and perhaps those are the things we can address. Good part is ATX-018 also can be efficiently manufactured. It's a single-step manufacturing. Both erythritol and phenylbutyric acid are very, very inexpensive starting materials. Now, both Ravicti and ATX-018 are currently not approved for SLC6A1, and either also for STX-BP1 or any other indication. And we would have composition of matter patent, but they would not. So I think it provides a case for any investor to come in that we could provide exclusivity as long as they're willing to work with us in terms of pricing and affordability of the drug. Now, I will skip through the next few slides. You've seen it before, so I don't need to show you, but I will stay with this one. This is from Dr. Kang's lab, where we actually have shown that ATX-018 works in her models. And it does have the same effect as phenylbutyric acid in her models. So this is very exciting. So in the sense, if you look at the prodrug of ATX-018, it works in the models as phenylbutyric acid. So it does the job, which means it is releasing phenylbutyric acid in those conditions. We have done PK studies in animals to show that ATX-018 releases phenylbutyric acid exactly as Ravicti, but at 25% lower dose. And Dr. Greenspan has shown that Ravicti works. So if you can see the translation that the dots are all connected, our main issue right now is to go in and demonstrate that it works. And I've been in this industry 35 years, and I can tell you this is about the best assurance you can get for a drug's potential to have efficacy in the clinic, because it has no safety issues, and you have all the correlate data that suggests that it should work. Again, this is for the science part that Dr. Kang has already shown, that PBA does have an effect on GABA uptake, so it not only reduces seizures, but mechanistically it does affect the GABA uptake in these mice. I will go through the STXBP side just briefly because there's a correlation that Dr. Greenspan mentioned in terms of its applicability. So a potential for ATX-018 is to do a similar cohort in SLC6A1 and STXBP1 as a proof of concept trial under an investigator-initiated study with Dr. Greenspan and Demarest. I'm going to skip the next two slides because you've seen it before. So where do we go for next steps? The steps as I've delineated are reasonably simple. So we need to do a pre-IND package that we will go in front of the FDA with the data we have today, and that we would suggest a potential for an investigator-initiated trial with the FDA, which is a trial that is a non-commercial trial, meaning you're simply looking for a proof of concept study. And one could start that filing within 6 to 9 months from project initiation. That does take care of all the other requirements that one would have to fulfill to file this pre-IND package. And then once you are done with the POC trial, you can take the data and go into a commercial registration trial to get the drug approved. So as I said, what's next? I mean, Dr. Kang and Dr. Gray they have shown the effects on PBA and the translocation, the GAT1 and GABA uptake. And as I said, a key component of mechanism of action is ER stress and the UPR, unfolded protein response, the UPR. Both of these components are tied into the mitochondria. And, one of the questions that intrigued me after hearing Kavanaugh's data today was that what is the phenylbutyrate doing? And there is a very clear evidence that endoplasmic reticulum and the mitochondria are tightly associated to these mitochondria-associated membranes. And this also has a role in a lot of other neurological diseases such as Alzheimer's. This alteration of the MAM, how it plays, has a significant role in how the progression of the disease or the function of the phenotype typical response to the patients. So one question that we've been intrigued about is what is the role of the mitochondria in this process? And question comes is the PBA acting as an anaplerotic agent? What this means is that is the PBA coming in as an external agent to support the TCA process? And perhaps it's not such an efficient molecule, hence you need a high dose. Now, if you think about this, this is not so out in terms of an extraordinary hypothesis, because when you have the patients that have each insult that is inherent in the system, the bioenergetics of those cells are definitely compromised. In that sense, the mitochondrial bioenergetic is now caught up in addressing the insult constantly, and it's falling behind. And that depletion or dysfunction in the mitochondria could be relevant to the story, or at least could be part of the story that can play out. And of course, since the ER and the mitochondria are tightly associated, one could definitely look at it, or at least explore the potential of it. So can we find a better anaplerotic agent? And perhaps some of the data that Dr. Kavanaugh is generating may suggest that, though I'm not sure that it's definitive. You will hear from him after me to understand his idea on that. But if that is the case, there could be other anaplerotic agents that are quite available that could work better than PBA. And perhaps that's a site of exploration we can look at. And the other component is that in conjunction with the PBA, can you use agents that can protect the mitochondria themselves? Because in this stress, there must be definite effect on the mitochondria in this particular disease. So we're starting to discuss this with Dr. Kang's lab and see if there's some ways to study these other agents or combination of agents, which are all clinically approved, they're known to be safe, and if we can find some ways to study those in the preclinical model, then I think we could have some ideas about some newer therapeutics outside of PBA. And last, I'll leave you with mitochondria is that we all know it's present in all human cells, of course, except the RBCs. They are associated with the genesis of life, and I do believe that in any pathology, mitochondria will be affected. There is no situation that I know of where the mitochondria are not going to be affected. And we have identified agents that really stabilize, protect, and induce biogenesis in the mitochondria that are already in the clinic. And one interesting aspect is that most therapies these days address the pathology and of the disease. And if we upregulate the mitochondria, perhaps you can induce a possibility of some healing because they go to the heart of the bioenergetics of the system. You're giving the system an option to heal itself.